Hi guys, now we quickly revise porphyria. It is a disorder when there is a defective in heme synthesis enzyme. All the porphyrias are autosomal dominant except CAXC. C for congenital erythropoietic porphyria, A for aldehyde deficiency porphyria, X for X-linked protoporphyria, E for erythropoietic porphyria. CAXC. These are autosomal recessive and X-linked protoporphyria is X-linked dominant. Okay, rest all are autosomal dominant. This is the pathway of heme synthesis. Succinyl-CoA glycine forms delta amino levulinic acid. This step happens in mitochondria. Now next few steps happens in cytoplasm. Last few steps again happened in mitochondria. Okay. In this step, allosynthetase enzyme is required, and for functioning allosynthetase, vitamin B6 enzyme is required. From delta amino levulinic acid, PBG is formed. From PBG, HMB or UPG1. From HMB, uroporphyrinogen 3, then coproporphyrinogen 3, then protoporphyrinogen 9, protoporphyrin 9, then ultimately heme. From delta amino levulinic acid to PBG, aldehyde enzyme or PBG synthase enzyme is required. As PBG is synthesized, so name is PBG synthase. From PBG to HMB or UPG1, HMB synthase or PBG deaminase or UPG synthase 1 is required. From uroporphyrinogen 3 to coproporphyrin 3, uroporphyrinogen decarboxylase is required. Wait, we remember these things by the help of some mnemonics. Okay. Now, from protoporphyrin 9 to heme, ferrochelatase enzyme is required. These are the important steps. Allosynthetase. Allosynthetase deficiency leads to X-linked protoporphyria. This is the X-linked dominant disease. This step is where? This is the step, first step. And this is the rate limiting step. Now, aldehyde or PBG synthase leads to aldehyde deficiency porphyria, this step. And in late poisoning, aldehyde enzyme is deficient. It is the major enzyme deficiency. In late poisoning, two enzymes are defective, aldehyde and ferrochelatase. Aldehyde is the major enzyme, ferrochelatase is the minor enzyme. Okay. Now, the third porphyria, HMB synthase or PBG deaminase or UPG1. This is caused acute intermittent porphyria. This step, HMB synthase, PBG deaminase or UPG synthase, acute intermittent porphyria. Acute intermittent porphyria is the most common acute porphyria. Mind it, acute porphyria. Clinical feature is neurocutaneous symptoms. Most common clinical feature is abdominal pain. Okay. Now, how to remember? Remember HSPD, HS, HMB synthase, PBG deaminase. Aldehyde is also called PBG synthase, so it is PS, but here it is PBG deaminase. So aldehyde is also called PBG synthase. HMB synthase also called PBG deaminase, also called UPG1 synthase, caused acute intermittent porphyria. Okay. Now UPG3 synthase leads to congenital erythropoietic porphyria. It is also called Gunther disease. This step, UPG3 synthase, is called Gunther disease. How to remember? C. C is the third letter, UPG3. That's how we can remember. And this is the picture of Gunther disease. Skin, teeth, these are involved. Now, UPG decarboxylase leads to Porphyria cutanea tada. This is the step. Uroporphyrinogen decarboxylase, Porphyria cutanea tada. This is the most common porphyria. Here photosensitivity is present, blister present. This point very important because it can differentiate from erythropoietic porphyria. Blister is present associated with hemochromatosis. This is the picture of Porphyria cutanea tada. Now how to remember? Here decarboxylase name contains C. Here the name contains C. Ferrochelatase, the last step, ferrochelatase. Here ferrochelatase. Ferrochelatase is the last step. This leads to erythropoietic porphyria. How to remember? E. Next letter F. F for ferrochelatase. It is the most common porphyria in children. Blister absent. In Porphyria cutanea tada, blister present, but in ferrochelatase deficiency means erythropoietic porphyria, blister absent. Erythema present, eczema present, photosensitivity present. This is the picture of erythropoietic porphyria. And lead poisoning leads to ferrochelatase deficiency, as I previously mentioned. So lead poisoning leads to aldehyde deficiency, also ferrochelatase deficiency, but aldehyde deficiency is the major one. Now we can quickly revise it. All the porphyrias are autosomal dominant. All the porphyrias are autosomal dominant except CAXC. C for congenital erythropoietic porphyria, A for aldehyde deficiency porphyria, X for X-linked protoporphyria, E for erythropoietic porphyria. Now these are the steps. Don't get scared about it. We have already simplified the steps. Now ALA synthase deficiency causes X-linked protoporphyria. Now aldehyde deficiency or PBG synthase deficiency causes aldehyde deficiency porphyria. In lead poisoning, it is the main enzyme defect. HMB synthase, also called PBG deaminase, also called UPG1 synthase, causes acute intermittent porphyria. Most common acute porphyria, neurocutaneous symptoms. Most common symptoms is abdominal pain. Now UPG3 synthase, congenital erythropoietic porphyria, also called Gunther disease. UPG decarboxylase causes Porphyria cutanea tada. Most common porphyria, blister is present associated with hemochromatosis, photosensitivity present. Now ferrochelatase deficiency causes erythropoietic porphyria. Most common porphyria in children, blister absent, erythema present, eczema present, photosensitivity present, and lead poisoning leads to defective in ferrochelatase, but it is the minor enzyme. These are some important points about porphyria. If you like this video, please like, comment, share and subscribe. See you in next video. Tata.